Hello and welcome to our May Publications podcast. I'm Ed Vital from the University of Leeds and chair of the Lupus Forum. And during this podcast, I'm going to review the recent lupus data and what it might mean for our clinical practice. Today, we've got 4 papers to share with you. And the first of those is actually a review article. This is a review article on the challenges of gaining FDA approval for CLE-specific treatments in the context of the phase 2 litifilimab studies in CLE and SLE. So this is quite an interesting trial program because, as we know, we've mostly developed drugs for SLE, but there are quite a lot of patients who have cutaneous lupus without systemic features. Probably a lot of these drugs would work for them too. They're just not licensed and proven in those contexts. So what Biogen tried to do when they were designing their program was put together a sort of set of trials that would cover both of these bases, CLE patients and SLE patients, and SLE patients with skin disease, some of them, or SLE patients with a lot of problems like arthritis. So that makes it's a relatively complex set of trials that takes a little bit of time to navigate through. But fortunately, one of our steering committee, Professor Victoria Werth, is an expert on cutaneous lupus and was also the chief investigator on some of these trials. So I went to her and asked her if she could give us her thoughts on this.
So the litifilimab trials, I should say, is really interesting because it's really the first time that there was a Part A and a Part B, with Part A being for SLE and Part B being for cutaneous lupus. And I think this is a really interesting and important model because who gets recruited and the outcomes that are used will be quite different for an SLE trial than a CLE trial. And this drug, as you likely know, has effects on plasmacytoid dendritic cells. And the way it works basically is you get internalization of the BDCA2 receptor on the plasmacytoid dendritic cell when the drug binds to this receptor. And as a result, we see a downregulation of both FC-dependent and FC-independent actions with decreased pro-inflammatory cytokines and chemokines. And in particular, pro-inflammatory cytokines that are decreased include IL-6, TNF-alpha, type 1 interferons, and type 3 interferons. So this is a very interesting mechanism, a very targeted mechanism. And I think what's really interesting is that the way that the development pathway was started was with a phase 0 trial, where essentially one could see the effect of only one injection subcutaneously in terms of the type 1 interferon expressed proteins in the skin, in particular MIX-A. And the effects were very quick. So there was demonstration of really a very good mechanism of action and also efficacy in many of the small number of patients that were looked at in the phase 0 trial. So in the Part A trial, which is an SLE trial, only a subset of patients had sufficient skin disease to be able to be really evaluated. And so if one did that, there was clearly a decrease in the skin activity as measured by the CLASI activity, with at least a 7-point drop in the patients who were treated in 56% of participants who got the drug versus 34% in the placebo group. And so this is a rather substantial difference, but again, not really powered to be able to statistically show that difference. In Part B, there were more doses that were examined, and it was possible to look at the doses, 3 doses relative to placebo. And it was very clearly demonstrated in a statistically significant manner that there was a decrease in the CLASI activity in all 3 doses relative to placebo. And the differences were very marked. The placebo response in skin was only about 14.5%. And it was more like between 39 and 48% in the 3 treatment arms over the course of 16 weeks. And so again, rather rapid improvement right initially in the first 2 to 4 weeks. And what's also remarkable is that the placebo response is quite low, and that probably reflects the fact that you don't need as much background meds in this population, and so you don't see as much of a placebo response. So overall, I think that this data is very encouraging and would make one really look forward to seeing the results of a larger phase 3 trial. I will also mention that the endpoint with the CLASI worked extremely well, and we know that what's an important difference in the CLASI-A from a patient perspective, and that the quality of life is quite improved when you see the kinds of changes that were seen in the Part B, the skin part of the trial. The other take-home message is that separation of SLE and CLE trials, but using the same drug, can be very powerful, again, because one can use a targeted endpoint for the organs that you're trying to measure. So overall, I think I'm quite enthusiastic about the potential efficacy of this approach, and we'll see. Stay tuned.
Okay, so there you have it, a review that tries to put together all the litifilimab studies. They're in phase 3 at the moment, so we're all going to be looking out for the results of that. I mean, my additional thoughts, I think one is that exactly how litifilimab works is becoming a more and more interesting question. Does it deplete PDCs and stop them producing interferon? Does it target PDCs and stop them doing something else? Or, in fact, as some people think, and I think, is that these drugs may even affect other cell types that are important in cutaneous lupus, like myeloid cells and macrophages. I think the other thing I think about in terms of how this goes forward, you know, when we've got these drugs in phase 3, obviously you just want to look out for the phase 3 outcome and find out whether this drug is going to make it into the clinic or not. But ahead of that time, I'm always thinking about, well, who are going to be the patients with the unmet need by that time? We've just had anifrolumab licensed. It looks pretty good for skin. We've got belimumab as well. Who are going to be the people where those drugs aren't suitable and this would be better? Or so that's always what I'm thinking about. So on to the next paper now. So this is a study that attempts an indirect comparison of belimumab and anifrolumab. So interesting here, you may remember that last year we reviewed another study that tried to do this. This was a paper by Ian Bruce and colleagues that tried to compare these drugs. And so this is a second attempt at a similar question. So the issue is you've got belimumab with a set of trials that show it's better than placebo. We've got anifrolumab with a set of trials that show it's better than placebo. Is one of those two drugs better than the other, or is one of them better for certain types of patients? And we don't have a head-to-head trial to tell us. So when you don't have a head-to-head trial, what can you try? Can you try and compare the individual trials you've got? Now that is obviously very problematic because certain people will go into one trial and not into another. They're done at different times. There might be some differences in design. There could be all sorts of other differences in the way the trial was done, what standard of care was used, how the outcomes were measured, all of these things. So there are a set of methods that try and solve this problem by doing what they call indirect treatment comparisons, where you take the data from the two trials. You can't just say, oh, the SRI-4 rate on the trial was this and on the other trial it was that. That's not going to work. But what you can try and do is see if you can account for the differences between the trials to get a better estimate. So this is the Bruce et al. Paper did this thing called a population adjusted treatment comparison. And they concluded that actually they thought the anifrolumab treatment effects were better than belimumab. So here, this is a different methodological approach to essentially similar trial data, not exactly the same, but similar trial data. And they've tried to do a comparison in a different way. And they make a few interesting points, if you read the paper properly, that they think there are some limitations to the previous analysis. So you have to be able to make one trial more comparable to the other. You have to kind of know what the main differences are and know how they affect the outcome. so that you can adjust for them. So, for example, if you had a higher SLEDAI in one trial than in the other, and you thought that was associated with the better response, then you try and make an adjustment when you compared those two trials. And that is clearly potentially fraught with a lot of problems, and you can see some of those problems that existed. Like, for example, there are some differences between two different trials of anifrolumab or two different trials of belimumab that themselves are not explained by these modifiers, these effect modifiers. So there are certainly things going on in these different data sets of all these different trials that we can't account for, we can't measure, and we don't understand. So you have to, any comparison is going to be a bit limited. So they got a different result when they tried to emulate the previous analysis published by Bruce et al. That they couldn't explain. They thought that it could be explained if maybe some of these effect modifiers, so effect modifiers are things like higher SLEDAI, more steroids, ethnicity of the patients, things like that. They thought that what if some of those measures had one effect in one direction on one of the drugs and an opposite direction on one of the other drugs? Could that occur and account for the differences? Well, they actually discount that possibility, but I'm not sure I'd be so hasty. I think it's possible. They also used some other statistical methods that are quite complex, that they use something called multi-level network meta-regression, that they thought made better use of the data. And in the end, their conclusion was that they couldn't really find a difference between the two drugs. So what does this tell us, and what do I think? So I think such comparisons of this are really too difficult to make any conclusion at all about. We can measure something in one trial and say, oh, that trial had a higher SLEDAI than that trial, so let's put in an adjustment for that. But what about all the things you can't measure? So, for example, outcomes in lupus patients, we've recently found in some of the work in my group, where we were looking at clinical outcome measures and biomarkers and things, trying to predict patients' quality of life. And we found that our sort of model didn't really work very well. And then we had a clever plan that actually maybe it was something to do with socioeconomic status, and we were able to adjust for that and also for the other medical conditions they had. And when you did that, suddenly the model became more meaningful. So that if you think about a lupus patient and when they get a good outcome or not, it's not just about their SLEDAI score and the treatment they're taking. They've got a whole life, and there's all these things going on that affect that, that are important to them. And so when you think, so belimumab trials done 2011, anifrolumab trials done 10 years later, what's changed in that time? Well, belimumab was licensed, of course. So there was another biologic on the market. We learned more things about using less steroids. Maybe there's some demographic changes in the countries. Maybe different sites took part. So, you know, there's that saying that you can't step into the same river twice. And I think that applies to clinical trials. So there might be all sorts of stuff that you don't know between these trials. And I think these comparisons are difficult. In terms of clinical practice now, we still don't have an answer to our question of, like, the next patient I see, should I choose belimumab or anifrolumab? And does it depend on a specific reason? I don't think we have a clear answer to that. We've got some hunches that maybe if they've got predominantly skin disease, you might think about anifrolumab. What about if they failed a lot of the B-cell therapies before? What about on their ethnicity? There may be some things like that, but I think we're learning as we go. Okay, so our next paper was one from the SLIC cohort. So obviously the SLIC cohort is something we cover quite often. It's this prospective cohort of patients who are taken from either at diagnosis or very close to when their diagnosis occurred. And what that lets you do is look at what determines which patients are going to get which outcomes. So this was quite a nice analysis that was based on antibodies. So what they did was they took 805 patients and they took their serum and did quite detailed antibody testing. So they did 29 ANA and immunofluorescence patterns and 20 different autoantibodies. And then what they did was use kind of machine learning methods on the antibody data. So in other words, just trying to say what sort of groups exist here of all these different antibodies. So they made clusters. And then they just looked at those 4 clusters to see how things turned out long term. And they got 4 different clusters. First one is this group of patients who had a high level of anti-Smith and anti-U1 RMP antibodies. So those antibodies are typically associated with quite severe immunology, at least. And in line with that, 10 years later, those group of people had highest damage, highest disease activity, more immunosuppressant and biologic use. The second cluster was a milder one. So this had a low frequency of anti-double-stranded DNA antibodies and a high frequency of these antibodies called DFS70, diffuse fine speckled 70. So those are antibodies that we think are often found in people with false positive ANAs. It's kind of a low-risk antibody. And the 10-year data, it was, again, consistent with that. So they had low disease activity, low likelihood of nephritis. Then you've got cluster 3 that's got a lot of antiphospholipid antibodies in it. And those patients also had, at 10 years, they had some interesting lupus features. They're more likely to have seizures, more likely to have low complement. And then cluster 4 was another quite severe group. So this is a group of patients who just had a large number of different antibodies. So quite a broad spectrum: histone, DNA, ribosomal P, Rho, LA, PCNA. So they had a large number of different antibodies, and they also got high disease activity. So what this is all about really is alerting you to the fact that when patients are diagnosed with lupus, sometimes on the day of the diagnosis, they're not that severe yet, but they are going in a severe direction. So you can spot people when you know this is potentially going to be bad. They might be all right now, but future there may be more problems. And you need to recognize that to be able to either at least monitor them more closely and probably treat them a bit more aggressively from the start. And this is just one of the ways that you can do that. So our last paper is one about ACR quality measures. So this is interesting because when I read this one, I thought, well, this is a bit unusual because I'm going to tell you something that's different to what I normally say. Normally, whenever I talk about lupus evidence and how to treat patients, I'm always saying things like, well, you need to take everybody as an individual and you look at exactly what's wrong with them and try and tailor what you're doing to their specific requirements. This is kind of the opposite philosophy. What if you were treating lupus like public health? What if you were saying, out of everybody with lupus, everybody in a country who had lupus, what should they all get? Like what interventions, if you could do it to every single person who's got lupus at a kind of public health level, and you might monitor that on some electronic health record that covered a whole hospital or a whole region, then what should that be? So that's what these ACR quality measures are for. So they're defined quality measures that they're meant to be things that you could extract from electronic health records to develop the measure in the first place, and also how you would use them and implement them. So there's quite it's very nicely laid out how they did it in the paper, of how they did this. They analyzed the different treatment guidelines that were out there. They developed some potential quality measures. They started with 57 of them. They narrowed it down to 15, and then they had a Delphi process where they tried to agree on the ones that were important and feasible, because there were some things in there, like, for example, what induction regime you get for lupus nephritis is clearly important, but they also found it quite difficult to extract from the data at a population level. So they ended up with 5 most important measures and 3 that they really recommended to be implemented. So those 3 measures were: 1, if a patient's got SLE, then they should have a prescription for hydroxychloroquine on or after the date of the most recent rheumatology visit, unless there's a contraindication or adverse event. In other words, give everyone hydroxychloroquine if you can. Number 2 was that the glucocorticoid dose should not exceed more than 7.5 milligrams of prednisone or equivalent for more than 6 months. And number 3 was that patients who've got SLE should have their kidney function and their protein excretion measured at least every 6 months. So they may sound like relatively simple statements, but the philosophy here is that very simple things that are implemented thoroughly and well can be effective when you look across whole populations of patients. And so I guess that's what it means for practice is, could you answer questions like these from your local databases, your electronic health records? Could you audit your practice and say, what percentage of my patients aren't meeting these basic measures? And so that you could put something into place to pick those people up and try and implement it. So that's all we've got time for today, and thank you for listening. All the papers we've discussed today are available as full slide sets that are free for you to access and download at lupus-forum.com. So you can download those and use them in your own journal clubs or your teaching. And if you register for updates on the Lupus Forum, you'll get emails whenever we have new content like this available. You can also follow us on Lupus Forum, or one word, on Twitter or on LinkedIn. Thanks again, and see you next time.